Hello students, as continuing with the same chapter and the immunity, today we are going to discuss about antigens. Antigens are the very important feature of the immunity. Antigens, these are the substances when introduced in the body stimulates the production of antigens. Next, these antigens are mostly conjugated proteins, means they are mainly proteinaceous in nature, sometimes conjugated with other molecule. That's why they are called as conjugated protein, like lipoprotein, that is a main core protein with a lipid molecule. Next, glycoprotein. Here the protein is conjugated with carbohydrate. And nucleoprotein. Here also the protein is combined with the nucleic acid. So the antigen may be like conjugated protein with any biomolecule, with a lipid, glucose, carbohydrate or nucleic acid. Next, the structure. The structure of antigen. If we discuss the structure of antigen, first is main important thing is antigenic determinants. Antigen determinants are called as epitopes. These are very crucial structural part of antigen. Antigen determinant or epitope are the components of antigen. Each antigen have many epitopes. These Y-shaped antibodies antibodies are Y-shaped in nature. So this Y-shaped antibody molecule at least has two binding sites that can attach to specific epitope on an antigen. So the antigen combines with antibody and form antigen antibody complex, and this complex will destroy the cell. This combination, antigen antibody combination, is very much like lock and analog. A particular lock has a particular key. Likewise, a particular antigen will have a particular antibody to kill or destroy the cell. So now to make more clear, we'll see the diagram. Suppose this is a cell. This is an antigen. This is the antigen. This antigen will have, these are the epitopes. As we have discussed, the epitopes are present on the surface of the antigen. So these are the binding sites. At this binding site, the antigen will bind with, this is anti, body, and these are the binding. These were the epitopes. Likewise, there are many epitopes on the surface of the antigen and the bindings for the binding of antibodies. This is the structure. This is the structure of antigen. These are the binding sites of both antigen and antibody. This is which will bind to a particular antigen and will now the types of antigen. If we discuss the type of antigen, here we broadly define two types of antigen: the complete antigen and the partial antigen or incomplete antigen. The complete antigen able to induce antibody production with a specific reaction. Means when any antigen will enter the body, specific antibody will be produced against it. While in case of partial antigens, they are called as haptens. Haptens are the substances which are incapable of inducing antibody formation by themselves. But when they combine large molecule, means the hapten, which are mainly the proteinaceous in nature, protein, which act as a carrier, which serve as carrier, will act as potent antigen, which have the capability to induce the antibody formation. There is also an antigen which is called as H antigen. H antigens are the antigens which are present on red blood cells of all ABO blood group. RBC of all ABO blood group contain antigen. Now, second topic. This is all about the antigen. The study is antigen presenting cell called as APC. Now, when the antigen is present, it will be destroyed by the different cells, how they are presented and persist. Antigen presenting cells are the cells that can engulf antigen and present fragment to T cells are called antigen presenting cells. There are mainly three types of APC in the body. The first is macrophage. Macrophages are phagocytic in nature, that is, they engulf the cell and greatly increased when stimulated to become active macrophage. They are present along the lymphocytes. In all the lymphoid organs and tissues. For example, monocytes as blood macrophage, histocytes as tissue macrophage. This is all about the macrophage. The second and most important we're going to discuss is dendritic cells. The next APC is dendritic cells. Give primary function as highly effective antigen trapping and antigen presenting cell, that is APC. These cells are non-phagocytic in nature. They are found in spleen, lymph node, thymus, and skin. There are different types of dendritic cells. Different types of dendritic cells. They are categorized on the basis of their presence. First is Langerhans dendritic cell, which is present in the skin, and trap the organism, come in the body surface, contact with the body surface. The second is the dendritic cells in spleen, which trap the antigen in blood. Next is follicular dendritic cell. They are present in the lymph node, trap the antigen in the lymph. Thus, these both macrophage and dendritic cell play important role in antigen presenting to T and B cells. These dendrite cells are having long cytoplasmic processes. So these are the processes. This is the nucleus. And these are the protoplasmic processes. Now the next APC is B cell. B cell express immunoglobulin molecule on the surface and function as antigen presenting cell. B cell descent, that is plasma, produces antibodies. This is all about the antigen and antigen presenting cell. Thank you.